Okay, this is the second half of the chapter on herpes viruses. In the first half, we went over classification and some of the clinical conditions caused by the various herpes viruses. In this portion, we're going to go through an overview of the molecular virology and replication, as well as just talk briefly at the end about latency, since that is one of the key hallmarks of herpes viruses. So as you saw in the first video, right, we had some pretty diverse clinical conditions caused by these family of herpes viruses. But despite the fact that they're causing different diseases in terms of the molecular virology and their structure, they do all have some similarities. So these are all enveloped icosahedral viruses, and they're all pretty big as far as icosahedrons go. And gets a little crazy, as you can see here, when you start looking at the structure, because there's a lot of an unusually large number of proteins that are produced by this virus and associated with the virion. So you have the capsid here in the interior that's made of 3 different major proteins as well as a handful of minor proteins. So we're not going to go into all the detail on that, but suffice it to say you have multiple capsid proteins making up that nucleocapsid. And then you can see this rainbow color of little circles here within the viral envelope. These are what are called the tegument or tegument proteins. And this can be as many as 40 different proteins, right, making up that population. And they're primarily within the virion in 2 layers. So you can see there's one layer just under the viral envelope, and then one layer associated with kind of the outer surface of the capsid. And there's so much of these tegument proteins, they estimate it may make up almost half of the entire mass of the virion. So there's a lot going on within the envelope, and then if you look outside the envelope at the surface there, you've got at least 11 different glycoproteins coming out of the surface of the virion. So it's busy, to say the least. Now if we look just at the genome, so this is a double-stranded DNA virus. It's linear initially, as we'll see when we get into replication cycle. It does get circularized as one of the first steps once we get into the nucleus, but within the virion, linear double-stranded DNA. And maybe not surprising, since I said these are large icosahedrons, the genome sizes can vary quite a bit, but are all fairly large for viruses. So the smallest in this group is VZV at over 100,000 nucleotides, and then the largest is going to be over 230 kilobases. So pretty big as far as viruses go. And of course there's going to be some differences in terms of the actual architecture, the organization of the genome, but there are some similarities that I wanted to focus on. They all have terminal repeats. That's what's indicated here as TR at the ends of the DNA genome. And then some, but not all, will also have internal repeats. And these are truly repeated sequences, although there are some actual sequences with specific functions like promoters, origins of replication, and occasionally you'll find some genes in those terminal repeats or internal repeats, but less common. And then those repeats are going to be interspersed with what are called the unique sequences, and those get designated either unique long or unique short, but that's primarily where you're going to find most of the protein coding genes in those unique sequences. Now, luckily we're not going to go through replication cycle for all these different herpes viruses. Some of them just haven't been all that well studied. The ones that were first identified, herpes simplex 1 and 2, have been the best studied. So we're going to kind of look at an overview that's based off of herpes simplex 1 or human herpes virus 1 as the model here. So we'll start with attachment, penetration, uncoating. I mentioned there was a lot of glycoproteins on the surface, and you hopefully got that idea from the cartoon that I showed you. But this always cracks me up. They estimate there's approximately 659 protein spikes. It's a very specific number, but many hundreds of glycoprotein spikes on the surface of the virion. And there's only a handful of those that seem to be key for attachment, at least in herpes simplex 1. And those have these like letter designations that you see here. So G for glycoprotein, B, C, D, H, L, etc. But there are two glycoproteins that primarily bind to what are called GAGs, glycosaminoglycans, on the surface of the cell, and those GAGs are primarily heparan sulfate and chondroitin sulfate that you see here in green and blue. And you can see glycoprotein B seems to be binding heparan sulfate, glycoprotein C primarily binding chondroitin sulfate. And then there's third glycoprotein here on the surface of the virion that's going to bind to specific cell surface receptors that are going to vary depending on the cell type. So if we're talking about immune cells, T cells, B cells, monocytes, that cell surface receptor is HVEM, herpes virus entry mediator, aptly named. If it's epithelial cells and or neurons, usually going to be nectin family of proteins as the cell surface receptor. And then there's a third, and I think you find this in a pretty wide variety of cell types, but you can get this modified heparan sulfate that can also serve as a specific receptor. So essentially you had the more generic binding followed by the more specific binding here. And again, this will vary based on cell type and can also vary, of course, based on which specific herpes virus you're dealing with. So here's just a little bit more detail on the fusion process. So you get the binding of the initial glycoproteins, and that's going to trigger a fusion of additional glycoproteins on the surface of the virion. And out of those, we think the glycoprotein H is likely the one that's going to mediate fusion of the viral envelope with the plasma membrane. So this is an instance where you're actually having fusion occurring at the plasma membrane for a lot of herpesviruses, although some can enter the cell by endocytosis. But I think this increase in size in the blue glycoproteins here is supposed to represent that fusion, with glycoprotein H as kind of the key player here. And they think that glycoprotein H is then undergo a conformational change that's basically going to pull the viral envelope into contact with the plasma membrane. Those two will fuse together, essentially creating a pore that then will allow all the tegument proteins and the nucleocapsid to come out into the cytoplasm. And you'll notice those tegument proteins that were surrounding the capsid continue to surround the capsid, and I believe in the cytoplasm they're going to help bind the nucleocapsid to the microtubule network for transport to the nucleus. So this would be, motor protein driven, primarily these two motor proteins, dynein and dynactin, which are going to drive movement of that whole nucleocapsid to a nuclear pore complex. There it's going to essentially uncoat, destabilize, and the double-stranded genome will get threaded through the nuclear pore into the nucleus, which is where nucleic acid replication is going to happen. And as we've seen with other viruses, this is an instance where you're going to get waves of transcription, right? So you'll have a first wave which will produce some proteins, but a group of transcription factors will then come back into the nucleus and activate transcription of a second wave. Those get translated. Some of those proteins are transcription factors which come back into the nucleus and initiate a third wave, and so on. So we have the immediate early genes that get expressed first, then the early genes, then the late genes. And you'll notice here the second wave, the early genes, a lot of those proteins are going to be key for replication, nucleic acid replication. And in the late genes, you're going to have more of the structural proteins, so capsid, those glycoproteins, etc. So again, we think this is really just to ensure that we get the virus genome replicated before we start assembling any virions. And in terms of the actual nucleic acid replication, it's really kind of a cool process. It goes through this rolling circle method of replication. And I mentioned upon entry into the nucleus, that linear double-stranded DNA genome is going to become fully circularized. The outer strand is going to get nicked in one spot. And then that 3' end there at the nick can basically serve as a primer for elongation, right? We would have then DNA polymerase come in and start elongating off that 3' end using the interior strand as the template. And you could kind of picture this whole thing just rolling as we go through the replication cycle, and you'll end up producing, tandem copies of the genome referred to as concatemers, that can then be individually packaged up into capsids, so you get multiple copies here being produced essentially at the back to back to back. Okay, so we crank out a bunch of copies of the genome there in the nucleus, and then this is also a little unusual. We're actually going to assemble the capsid inside of the nucleus. And remember, it's primarily that late group of proteins that are going to be the structural proteins that we need for this process. And I always picture like a little construction crew almost building these, because you actually apparently have some like scaffolding proteins that are involved with this whole process. Those are going to get removed by a virus-encoded protease. And the bottom line is you're going to end up with your assembled capsid with the genome inside, right, with tegument proteins surrounding it inside the nucleus. And obviously, we need to get that out of the nucleus. So we're going to go through this process called double envelopment. You may remember the nuclear envelope is a double layer of membrane. So you're going to see the capsid bud through the inner layer. So here's nucleus, inner membrane, outer membrane, space between, and then cytoplasm out here. So capsid's going to bud through the inner membrane, right? And then as it buds through the outer membrane, or fuses with the outer membrane, it's going to dump the nucleocapsid and the tegument proteins out into the cytoplasm. And then we're going to actually go through the ER to pick up what's going to become the actual viral envelope. We've seen this with other viruses as well. But you'll notice some of those glycoproteins, right, that were being produced have already localized to could be either Golgi or ER membrane. I'm not sure how the cell decides, maybe depending on cell type or particular herpes virus. But you'd have those glycoproteins on one of those two membrane surfaces. That's going to attract the virion, which will bud into the Golgi or ER, and then as it buds out, right, it's got what's going to be its envelope, and then an extra layer of membrane that will be shed when it fuses with the plasma membrane through exocytosis. And what gets dumped out is our enveloped virus there. So not too bad. I think this is, one of the cooler ones in terms of replication strategy. There's some unique features of at least herpes simplex 1, which again is what this was based on. So, hopefully you've gotten the idea. This whole idea of latency is key for herpes viruses. So how does that happen, right? How do we go through these cycles of activation and latency? And of course, the hallmark of latency is you're not actively producing any new virions, and it seems like you're really not making any virus proteins. But what they have found is, while in that latent state, at least herpes simplex 1 and 2 crank out a lot of what are called latency associated transcripts, which actually don't code for any proteins but get cleaved into what are called microRNAs. So these are small, short RNA molecules. So here's your viral genome, right? So here's our latency associated transcript getting cleaved into the microRNA. That can then leave the nucleus and associated with this little protein complex called RISC, RASC, which is short for RNA-induced silencing complex. And that microRNA gets loaded into the little RISC protein complex, and that then goes off and looks for any viral messenger RNAs that might have been produced, right? So presumably there is actually some transcription happening while in the latent state, but these microRNAs with the RISC are going to target those out in the cytoplasm for degradation. So that's going to prevent any translation of viral proteins from happening. And then I guess, at some point, for whatever the trigger might be, we will stop producing these latency associated transcripts, which is then going to let viral proteins be produced, and that's going to trigger the outbreak or little relapse of viral symptoms.